Assalamualaikum and hi, good morning to everybody. My name is Sarah Safia binti Zainal. My title for my research review paper is Bioactive Secondary Metabolite from Arctic Fungi. Introduction. Research background. Like the animal and plant which occur in the Arctic, polar fungi have evolved physiological and morphological characteristics to aid their survival. Instant cold heaviness in the polar desert and ultraviolet radiation from the sun is where the Arctic fungi can be found. A number of secondary metabolites have been described from Arctic fungi, isolate that exhibit many biological activity such as antimicrobial and antifungal. Problem statement. How some living such as fungi can survive in Arctic conditions. Second, most of the research provide information on the biological activity of fungi from terrestrial and tropical fungi, and very little work on fungi from polar environment such as the Arctic. So, the objective is, the first one is to elaborate on the adaptation of fungi in Arctic conditions, to review the biological activity of Arctic fungi, the third, to review in biological activity of antibacterial and antifungal, and the last one is to review methods of how to get the compound from the Arctic fungi. Significance of study. The study is essential to clarify the benefits and the special characteristics of Arctic fungi that other fungi are not able to have. The ability of fungi to tolerate high or low temperatures, which can actively live and tolerate such an extreme environment. So the research question in my research review is, what compound that found in Arctic fungi for biological activities? Arctic fungi can be found in soil or marine. So what is the important Arctic fungi in soil? It plays an important role in development and maintenance of the soil microbial community in the Arctic tundra, and it plays a vital role in regulating the energy and mineral cycle in the region. So the role of marine fungi is, marine fungi in fungal breakdown and the consequence of this metabolism on higher trophic level. And then marine fungi are major decomposer of woody and herbaceous substrate in marine ecosystem. This is the type of biological activity that I found in my research. The first one is cytotoxic activity. Cytotoxic is a biological activity defined as chemical potency that bring variation in cellular functions and result in cell death. The second one is antibacterial activity. It's a molecule ability to fight bacteria and viruses solely dependent on its ability to include substances that selectively. The third one is antioxidant activity. It's the restriction or prevention of nutritional oxidation, particularly that of lipid and protein. And anti-proliferative activity is the result of gene expression, regulation, and initiated by signal transduction, pathway, and transcriptional activation. This is some fungi that I found and I list out from cytotoxic activity, as you can see from my table. Next one is, this is some fungi that I found and I list out from antibacterial and antifungal activity, as you can see from my table too. This is some fungi that I found and I list out from antioxidant and anti-proliferative activity, as you can see from my table. Structure type derived from antibacterial and antifungal activity. Arctic fungus Nectaria, species B13. This is the compound that shown, the compound from the fungus Nectaria, species B13. And it show on the compound 4, show antibacterial activity against E. coli, B. subtilis, and S. aureus. Net is the compound from Arctic fungus Ethiopia species D1, and the compound 2 display antibacterial activity against S. aureus and E. coli. The antifungal activity of compound 1 until 4 against C. paraplesiosis, C. albicans, C. glyptera, C. neophorum, M. gypsum, and C. tropicalis. Net is compound from Arctic fungus Cochlobolus gusanoi. It's shown all compound has ability to screen the antibacterial activity to inhibit S. aureus, E. coli, and C. albicans. Method that used for find compound on antifungal and antibacterial activity. Chemical methods. First, alcohol. Alcohol are membrane-active microbicides, something universally acknowledged, whose antimicrobial efficacy. Ethylene oxide is alkylation reaction to cellular component of organism like nucleic acid and functional protein. Producing their denaturing is related to its microbiology inactivation capability. Next, formaldehyde. It is an electrophysically active microbicide having the capacity to react with a variety of amino acids found in the microbial cell, including purine and pyrimidine group of both DNA and RNA. Next, as an antifungal, membrane-active microbicide that fall under the imidazole or triazole classification, depending on how many nitrogen atoms there are in the azo ring. Next is essential oil, are complex volatile compounds synthesized naturally in different plant parts during the process of secondary metabolism. These are advantage and disadvantage of chemical methods used in secondary metabolite, as shown in the table. Conclusion. Adaptation from Arctic marine fungi is the most found secondary metabolite for biological activity. Antimicrobial and antifungal activity is the most biological activity found in this research for Arctic fungi. Recommendation. So, I will suggest to scientists to make research more on other biological activities other than antibacterial and antifungal. A true investigation should be carried out to determine whether more compound get from secondary metabolites.